Today, I would like to give you a brief introduction to our new analysis package F-scope for quantitative isotope analysis. To start, I would like to tell you a little bit about our company. We were established in 2003 in collaboration with Keio University. Our platform is based on capillary electrophoresis mass spec, and we have developed a proprietary data analysis program. We have a large library of over 1,200 compounds, where 900 are annotated and over 100 are accurately quantitatable. We can provide complex statistical analyses, and we provide you with a comprehensive pathway analysis. We have had a number of pharmaceutical and academic collaborations both in Japan and here in the US. As I mentioned, our technology is based on capillary electrophoresis mass spec, whereby compounds are first separated based on their charge and molecular size. An electric field is generated by applying a voltage to both ends of the capillary. In this schematic, we are separating the positive or cationic compounds, but a reversal of the charge applied to the capillary will separate the negative or anionic compounds. The compounds are then further separated based on mass and time in either time of flight or triple Q mass spec. Concurrent separation of hundreds of metabolites present in assay samples is achieved through these differences in mobility of ions. There are several advantages to adding a CEMS platform for metabolite profiling over the traditional LC or GC analyses, including higher resolution, a higher sensitivity for selected analytes, a unique metabolic space versus LC or GC, a faster analysis time, improved reproducibility and low relative standard deviations, simultaneous accurate quantitation, and our HMT data analysis software. Here you can see the low relative standard deviations, or RSDs, achieved as a result of measuring a standard solution 6 times. Typically, RSDs are at 2% or below and are similarly as low for anionic compounds. RSDs for LC analysis are typically in the 15 to 25% range. This allows you to be confident that any differences observed are real and a result of biological variations and not the technical platform. No single platform is capable of measuring the entire metabolome. While LC is particularly suited for the more nonpolar and hydrophobic compounds, and GC is more suited for the volatile organic acids, CEMS is best suited for the detection of the majority of small polar metabolites. In particular, CEMS is capable of detecting the majority of the metabolites involved in cellular metabolism, in particular the TCA cycle intermediates, phosphosugars of glycolysis, and the pentose phosphates, which are involved in aerobic glycolysis, commonly upregulated in cancer cells. In addition, though LCMS has methods for detecting nucleotides, these compounds are particularly suited for analysis by CE. Here you can see a comparative nucleotide analysis performed using CEMS and LCMS, and we show cleaner separation and better peak shape were achieved using CEMS, as well as a 40% increased detection sensitivity, which allows for better quantitation, accuracy, and reproducibility. Here we are looking at our dynamic range for one of our cancer cell studies. We can see over 5 orders of magnitude of coverage of the various metabolites. Knowing the dynamic range of the different metabolite levels allows us to provide accurate standard curve coverage for better quantitative analysis. Steady-state metabolomics provides insightful snapshots of cellular metabolic pools. Comparisons among samples can often reveal differences in glycolytic output or proliferation rates. However, due to the fluidity of metabolic processes, often subtle changes in preferred metabolic pathways can be missed. Heavy isotope flux analysis, which uses substrates including 13C or 15N, provides insight into the direction and flow of those labeled markers, typically glucose or glutamine. For glycolysis, lactate, the final product, is considered as an indicator of glycolytic output. But information from steady-state metabolic levels is limited because it reveals only the balance between production and consumption. Labeling analysis using 13C glucose can quantify the production derived from glucose, which is a direct indicator of pathway activity. Heavy isotope analyses can also often be used to distinguish between multiple enzymatic reactions that result in the same product. For example, citrate can be formed from the traditional oxidative reactions of the TCA cycle. However, it can also be formed from a reversal of the TCA via reductive carboxylation. Unlabeled steady-state metabolomics cannot definitively determine the directionality of the TCA. However, by using uniformly 13C labeled glutamine, a clearer picture can emerge, as one carbon will be lost as CO2 via the oxidative pathway, while all 5 carbons will be retained via the reductive pathway. Therefore, analyzing the change in percentage of 4 or 5 labeled carbons in citrate can give clues to changes in the directionality of the TCA cycle. These approaches can identify specific sensitive reactions for potential therapeutic intervention and mechanism of action studies. Due to our high-resolution technology, we can accurately detect and quantify the various isotopomer distributions using our F-scope platform. Our F-scope platform includes the quantitative analysis of 54 metabolites central to energy metabolism. Targeted metabolites in F-scope include those from glycolysis, TCA cycle, pentose phosphate pathway, adenosine phosphates, amino acids, urea cycle, and glutathione. Typical substrates include glucose and glutamine. However, other substrates such as N15 amino acids or succinate can also be used. Included in the analysis is a pathway-based visualization of isotope distribution and activated metabolic pathways. Sample requirement is low while maintaining a large dynamic range. Now I'd like to provide a few examples of how F-scope has been used by some of our collaborators. Recently, constitutive stabilization of NRF2 has been reported in many human cancers due to genetic or epigenetic reasons, including somatic mutations in KEAP1 or the NRF2 gene, accumulation of aberrant proteins disrupting the KEAP1 NRF2 interaction, reduced expression of the KEAP1 gene due to its promoter methylation, or modification of KEAP1 cysteine residues by oncometabolites. In all cases, the degradation of NRF2 is impaired and NRF2 is stabilized. Because NRF2 induces many cytoprotective genes, NRF2 positive cancers are expected to be resistant to chemotherapy and radiotherapy. In addition to promoting chemo and radio resistance, NRF2 strongly promotes cell proliferation, as a significant decrease in cell proliferation was observed after siRNA-mediated knockdown of NRF2. Using ChIP-seq, the authors show that NRF2 directly interacts with and modulates genes involved in the pentose phosphate pathway and NADPH production, including glucose 6-phosphate dehydrogenase, transketolase, transaldolase, phosphogluconate dehydrogenase, and malic enzyme 1. Because NRF2 directly activates those metabolic genes, the authors examined whether NRF2 alters the metabolite flow. These cells were pulsed with C13 glucose or glutamine. When C13 glucose was used, you can see a clear increase of glycolytic intermediates and a decrease of inosine monophosphate, the precursor of de novo purine synthesis. When C13 glutamine was used, you can see the reduction of lactate and glutathione accompanied by the increase of glutamine. This suggests that NRF2 promotes both glutathione synthesis and glutaminolysis, leading to lactate production, as well as de novo purine synthesis. A more detailed analysis interestingly revealed that when NRF2 is deleted, while IMP and PRPP were reduced, resulting in a decrease in nucleotide synthesis, the other pentose phosphate pathway metabolites were all increased, suggesting a stagnation of the pentose phosphate pathway. We can also see that there is increased flux towards serine as a result of the elevated glycolytic and pentose phosphate pathway intermediates. To confirm the metabolic studies, NRF2 was overexpressed and again subjected to the labeling experiment. We can see that after only an hour, C13 flux to lower glycolytic and pentose phosphate pathway intermediates was reduced with a corresponding increase in IMP. We can also see now that flux to serine and glycine synthesis is also reduced. In summary, NRF2 redirects glucose and glutamine into the anabolic pathways of purine and glutathione synthesis. So while oncogenic pathways involving p53, HIF, and mTOR facilitates glycolysis, and c-MYC promotes the uptake of glutamine and the conversion of glutamine to glutamate, we see that NRF2 helps fill in the gaps to push metabolites into the pentose phosphate pathway and towards glutathione synthesis. Next, I would like to briefly introduce our study on the mitochondrial complex 1 disorder MELAS, or mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, which is caused by a single mutation in a mitochondrially encoded enzyme for complex 1 of the electron transport chain. Patients with MELAS accumulate lactate in their plasma. To reduce acidosis, clinicians use DCA, which acts to enhance pyruvate conversion to acetyl-CoA, thus reducing lactate production. Pyruvate treatment, on the other hand, improves multiple symptoms, but the mechanism has not yet been elucidated. After addition of pyruvate to the MELAS cells, the metabolic profile shifted over to that of the control cell lines, suggesting pyruvate, but not DCA, restored the MELAS cells to a more normal metabolic phenotype. Using C13 labeled pyruvate, the authors showed that the added pyruvate was not converted directly into citrate, but instead the label was detected in alanine, along with an increase in unlabeled 2-oxoglutarate levels, suggesting that the alanine transaminase was activated with pyruvate treatment. 2-oxoglutarate can then go on to stimulate the TCA cycle, thus alleviating many of the symptoms associated with MELAS that simply removing lactate cannot. A number of publications have featured our isotope analyses along with what was presented here. Please see our website for links to these and other HMT publications. Thank you for your time, and please feel free to contact us or visit our website for more information.